In this Medicoapps Masterclass, we will learn about Lac Operon Concept. Now, before we go into learning of Lac Operon Concept, let's look at the structure of Lac Operon. So it has various genes as a part of it. Let's look at each of these segments. So first we have got is Lac I gene. Lac I stands for Lac inhibitor gene. Now this is a housekeeping gene. We will learn about the role in a while. So this is Lac I gene. Then we have got the CRE segment. CRE stands for cis regulatory element. Okay. So we have got this CRE segment. Now this CRE segment in very simple terms act as a positive regulator for the promoter. Okay, then we have got the promoter sequence. Promoter sequence are that sequence on which the RNA polymerase will bind and which will lead to ultimate transcription of all the genes. Then we have got the operator segment. Now operator is a short region of DNA that lies partially within the promoter and it interacts with the regulatory protein that controls the transcription of the operon. So it is basically the part which will interact with the regulatory protein. Okay, and then we have got the three genes which are a part of the Lac Operon, that is Lac Z gene, Lac Y gene and Lac A gene, and the transcription of these genes will ultimately produce enzymes which will lead to metabolism of lactose as a source of energy. So this is the entire structure of the Lac Operon. Lac I gene, CRE, promoter sequence, operator sequence, Lac Z, Lac Y and Lac A. Now before we move forward, let's understand another very important concept, the concept of polycistronic gene. Now polycistronic gene is a part of seen in prokaryotes. Now what it essentially means is that in prokaryotes multiple genes are under the influence of a single promoter. Okay, multiple genes, like in this case you can see Lac Z gene, Lac Y gene and Lac A gene. All these three genes are under the influence of a single promoter. So it's like a switchboard. So imagine a switchboard where one switch can open your light, can open your fan and can open your refrigerator, right? So this is a concept of polycistronic gene. In eukaryotes we have got monocistronic gene. Monocistronic genes means each gene will have its own promoter sequence. So tubelite will have its own switch which can be on and off. Then fan will have its own switch which can be on and off. Refrigerator will have its own switch on and off. So this is a very important concept of polycistronic gene as well as monocistronic gene. Now let's look at why this operon concept is important. It is a mechanism by which genetic expression is controlled. Okay, and why this genetic expression has to be controlled? We have seen that all these three genes, if they are transcribed, they have a common function that they help in digestion of lactose to produce energy. So the bacteria has a huge task when it comes to metabolism, that there are two sources of energy, glucose and lactose, and it has to decide that if it wants to use lactose as a source of energy. So in whichever condition it will wants to use lactose as a source of energy, in that condition these three set of genes should be transcribing. So imagine a normal condition when glucose is present and lactose is not present. So in this case obviously the bacteria does not want to produce these genes, so this Lac Operon will be shut down. Again, imagine a situation where glucose is not there but lactose is there. This is the condition where the bacteria wants to pump up these three enzymes so that it can utilize all the lactose available for production of energy. So in this case the Lac Operon will be switched on. Imagine a condition that both glucose and lactose is absent. Okay, then obviously in that condition also the bacteria does not want to switch on these genes because there is no lactose in the system, so producing these enzymes will be unnecessary. And the last situation is, imagine a situation where both glucose and lactose are present. In that case it will be switch on, but it will transcribe at a very slow rate, because when bacteria is there, because glucose is a more efficient source of energy, it will always prioritize glucose as a source of energy. So even in this case, when both is present, Lac Operon will be switched on, but it will transcribe in a very slow manner. Okay, now this is what the bacteria wants to achieve through Lac Operon. So let's look at how this entire thing is achieved. So first, as a part of housekeeping gene. So LAC-I is a housekeeping gene. Okay, housekeeping genes are those genes which keep transcribing under normal circumstances. Okay, so LAC-I gene in this operon structure is a housekeeping gene. So under normal circumstances it will produce, LAC-I stands for LAC inhibitor. Okay, it will keep on producing inhibitors which will go and attach itself to the promoter sequence. So what will happen if these inhibitors are attached to the promoter sequence? What will happen is it will not let the RNA polymerase bind here. Okay, it will inhibit the RNA polymerase from binding here, and so these three genes will never be transcribed in such a condition. Now imagine, so this is what happens in the normal circumstances when lactose is not there in the system. Now what happens is, imagine a situation when lactose comes. So lactose will bind to this inhibitor and displace it from the promoter sequence. Okay, so now when the lactose comes, it will bind with those inhibitors and displace it from the promoter sequence. Now what happens? The moment promoter sequence is free, what will happen? RNA polymerase can come and bind here, and then all these three genes, LAC-Z gene, LAC-Y gene and LAC-A gene can be transcribed. Now LAC-Z gene is for beta galactosidase protein, and the major function of that protein is to break down of lactose. LAC-Y gene is for permease. Its major function is for entry of lactose, and LAC-A gene is for thiogalactosidase, and majorly again it functions for entry of lactose. So now this promoter, so the moment lactose came, the promoter sequence now is free and it can transcribe all these three genes. Now let's bring glucose into the equation. So what happens? First we have to understand the concept of the relationship between glucose level and cyclic AMP level in the cell. So what happens whenever the glucose level is falling down, the cyclic AMP level in the cell will rise, and it's very simple concept that if glucose level is down, the level of ATP will be down. If the level of ATP is down, the level of ADP will increase, okay, which will lead to the level of AMP increase, okay, and which will be finally converted into cyclic AMP. So glucose and cyclic AMP level in bacteria is inversely proportional. So in a condition where glucose is absent or very less, the level of cyclic AMP will start rising. Now let's look at what this increased level of cyclic AMP does. So what does cyclic AMP does is, this cyclic AMP combines with a catalyte activator protein. Okay, and both of these will form a dimer, and what this dimer will do, this dimer will go and attach itself to the CRE segment. Now as I told you, this CRE segment is like a promoter to the promoter sequence, so it will activate the promoter sequence, and now the promoter will very readily bind with any RNA polymerase that is available in the system and quickly start transcribing all the genes. So when the glucose is absent, now promoter is really active, and if it is free, which means if the lactose is there in the system, it will pump all its enzyme to produce this LAC-Z, LAC-Y, LAC-A gene proteins, right? So this is what will happen when the glucose is absent. So when the glucose is absent, the promoter sequence is really pumped up because of the activation of this CRE gene, because of this dimer CAP and cyclic AMP dimer which is formed. So another very important question is, what is this catalyte activator protein? So this CAP acts as a positive regulator protein. Okay, so if catalyte activating protein is there along with cyclic AMP, it will act as a positive regulator for the Lac Operon system. Now what happens if glucose is present and lactose is also present? So this is the last condition. When glucose is also present, lactose is also present. When glucose is present, then this CRE will not be activated, and so the promoter will not be activated. So even though RNA polymerase will bind here, but the rate of transcription of these genes will be very, very slow. So this is what you have to understand here, that if glucose is present and lactose both are present, then because lactose is present, the promoter sequence will be free. Okay, so RNA polymerase can bind and can produce all these three enzymes. However, because the CRE segment is not activated and that CRE segment does not activate the promoter sequence, the rate of transcription of all these three genes will be very, very less. So in a nutshell, if I have to tell you, if glucose is present, Lac Operon is turned off, and if glucose is absent and lactose is present, then only the Lac Operon is turned on and functioning at the full level. So this is how the entire system is modulated so that bacteria chooses to use glucose when it is present, and when glucose is not there and lactose is present, it uses lactose as a source of energy. A very important term here is gratuitous inducer. So lactose analog capable of inducing Lac Operon are called as gratuitous inducer. For example, isopropyl thiogalactosidase. Okay, so if there is in the system isopropyl thiogalactoside, then what will happen? It will act as a lactose analog, and then again it can induce the entire Lac Operon system. So this is the entire concept of Lac Operon system.